Hello, I welcome you all to TrueNat Hybrid Training. I am Dr. Keerthivasan, microbiologist from Intermediate Reference Laboratory, Port Blair, of National TB Elimination Program. So we'll have this particular training in three parts. Today we'll have part one. So let's see what all we cover in part one. So first we'll see the various lab tests which we do here under our program. Then we will see something about PCR Basics because these tests are also based on that principle. Then we'll see what TrueNat really is, and what are a TrueNat site requirements. Then master checklist, that means what all things do you need to do a test. And then we'll see a complete sample workflow, that is how a sample is processed in TrueNat and how you get a result. Then we'll have post-test. So first thing first, so we have these centers called TB diagnostic centers. Okay, so TB diagnostic centers, so all our peripheral health facilities, most of the place we have the facilities of X-ray and microscopy. So when a patient who has symptoms of TB presents to a clinician, so clinician does a symptomatic screening or a clinical screening. So there we see whether the patient has some characteristic symptoms of TB. So that is our 4S screening. Remember, 4 symptom screening of TB. So what are the 4 symptoms? We have fever, cough, weight loss, and night sweats. So mostly of the duration of 2 weeks or more. Okay, so based on these symptoms, a clinician suspects that this particular patient may have TB. So he writes these tests which are available in his peripheral health center. So that is X-ray. So X-ray will give a picture of TB, but it cannot confirm. TB is caused by a bacteria, Mycobacterium tuberculosis. So X-ray cannot tell anything about the bacteria. It can give a picture, and that picture, the problem is there are other conditions which mimic the same picture. So we need something called a bacteriological confirmation that this is present and this bacteria is also present which is causing the disease. So we move on to our next test, that is microscopy. So most of you are aware of this microscopy staining technique, that is acid fast staining. So other than that, we also have another technique called fluorescence staining where we use oromin-phenol. So fluorescence staining we have in our district headquarters, whereas all in our peripheral centers we have acid fast staining. So once we have diagnosed, the clinician has diagnosed this particular patient has TB by bacteriological confirmation by staining, then what is the next step? The next thing test which we do is NAT test. So we have NAT centers. What is this NAT? Nucleic acid amplification test. So this is done with the help of two machines, TrueNat and GeneXpert. Today we will be discussing about TrueNat. So what additional information does it give? So it gives genetic confirmation that this particular bacilli which you are seeing is Mycobacterium tuberculosis. Because what happens if the LT is not trained, or he may confuse this bacilli which he is seeing in the staining, and he may give it as Mycobacterium tuberculosis, whereas there are chances that it may be something else. So we need a genetic confirmation, so that is given by this particular test. And not only that, it gives one additional information. It gives a resistance status of one of our first line drugs, that is rifampicin. So it gives a result of rifampicin resistant detected or not detected. Now, why is this important? In olden days, what we used to do, based on these two particular tests, a patient was put on treatment. Okay, so that simpler treatment, that is our drug sensitive treatment. Drug sensitive TB means the patient is sensitive to first line drugs. Okay, that we have isoniazid, rifampicin, pyrazinamide, ethambutol. So that course is simpler, and the prognosis, that is the chance of that person curing, is also quite high. And that duration is only 6 months. But what happens sometimes that bacteria which is present in the patient may become resistant to these drugs. These 4 drugs, these are first line drugs. And then if they are resistant, then these drugs won't work. So that time that patient becomes a drug resistant TB patient. So for them, another set of therapy has to be given, which prognosis is not that good and compliance is required. That means the patient has to take drug now not for 6 months, but for a longer duration and more number of drugs for him to be cured. So there is a delay, isn't it? So if we start everybody on drug sensitive treatment and later if there is clinical deterioration, where the patient is not responding and his disease has spread to other regions, then only we suspect he may be drug resistant. So it was causing a lot of morbidity. Okay, so that is why initially itself now these tests are available. As soon as your microscopy is positive, or even upfront, we do this test and we come to know about the status of drug resistance. Here, the drug which we use is rifampicin. If the patient sample has rifampicin resistant not detected, then we give first line treatment, that is the 6-month treatment. And if the rifampicin resistant is detected, then we consider this patient for drug resistant TB treatment, which is for longer duration. So these tests are quite important in modern scenario to start which particular therapy for that particular patient. Okay. Now coming to the facilities which are available. So these TB diagnostic centers in South Andaman, as you can see the breakup here in our 3 blocks or tehsils, Port Blair, Ferrarganj and Little Andaman. So we have total 10 centers here. NAT centers, again in Port Blair we have 3 machines. In Ferrarganj and Little Andaman we have proposed. So as our country is moving towards NTB goal, so our government has envisioned that all the blocks, all the tehsils should have this particular facility. So that is why we will be expanding this particular test to all the tehsils. Now coming to North and Middle Andaman districts, so here we have TB diagnostic centers, 6 are there. And NAT center in Mayabanda we have 2 machines, in Diglipur we have 1 machine, and in Rangat it is proposed. Now coming to Nicobar district, we have 4 TB diagnostic centers, and NAT machines we have in Car Nicobar and in Nankauri 2 each, and in Great Nicobar we have planned. Now let's compare all these tests which we have been using for screening and detecting TB. So first I told is X-ray. So sensitivity is quite good, 70 to 90%. That means the picture which resembles TB, it can pick up. Okay, so it can pick up the TB picture. But the problem is specificity. To tell that this particular thing which you are seeing in your chest X-ray is due to TB, because I told you there are certain other diseases and condition where the chest X-ray will look similar. So it cannot tell it is confirmatory TB, because for that you have to detect the bacteria. So that is why specificity for chest X-ray is quite low, 65%. Now coming to our bacteria detection. So first we have acid-fast staining. Okay, so this staining, acid-fast staining, is there since the time Robert Koch discovered our Mycobacterium tuberculosis. That is when 1882. Okay, so now it's 2022. So nearly 140 years. That is how old this particular staining technique is. So there you can see the sensitivity is from 20% to 80%. Why? Because it depends upon, in the particular sample you have given, how much bacteria is there. It is dependent upon the bacterial load. So it will give positive only when each ml of your sample contains 10,000 colony forming units. Okay. So only when if it is up to that much concentration of bacteria in each ml of a sample, this particular test will give positive. So there is a lot of chance that you may miss the diagnosis of TB if the concentration is less. Say for example, the initial stage of TB where the patient is coming with some symptoms, whereas the bacteria has not multiplied enough, so the number is quite low. So you may miss the diagnosis of this particular patient. So now next coming to fluorescence staining. So here we use little more sensitive technique. Fluorescence stains are used. So it is little better, 10% much better than acid-fast staining. Now we have our 2 tests, NAT test, Gene Expert and TrueNat test. So here you can see Gene Expert has around 98% specificity. Okay, so 98% confirm it can tell that this particular thing is TB and rifampicin resistant status. So here you can see there are different versions of these machines, the modules. So here for Gene Expert, most of the machines which we have here in Andaman Nicobar Islands are 4th generation, version 4. So they can detect as low as 131 colony forming units per ml. So here you can see staining 10,000 colony forming units, here just 131. So TrueNat, the MTB chip which we currently use, it can detect 100 colony forming units per ml. Both these machines have little more sensitive chips or cartridges also. So for example, Gene Expert has the Ultra version which can detect as low as 16 colony forming units per ml. And TrueNat also has a sensitive chip called MTB Plus which can detect as low as 30 colony forming units per ml. These are more sensitive because in the other 2 versions, they target only one region. So here in Ultra and MTB Plus, they target 2 regions. So they can pick up the bacteria with as low as 16 to 13 colony forming units. Now, before any test comes to the market for patient use for our healthcare use, these are tested, isn't it? Obviously. So, WHO conducted a multi-center study along with FIND. So, they did this study in almost 19 clinical centers in 7 reference laboratories and across 4 different countries. Okay, the study was done and it was compared. This particular or new test TrueNat was compared to the gold standard. The gold standard is culture. So any new test or any upcoming tests are always compared to culture. Okay, so here obviously as you can see, they saw a specificity in all patients very high, more than 95% specificity, and you can see all the patients where smear sample was positive, sputum smear positive. So here you can see the sensitivity is quite high. Sputum smear negative patients. So obviously sputum smear is negative. That means the number of bacteria in the particular sample is quite less. So here obviously the sensitivity is less. Not only with culture, they also compared with our existing machines for NAT, that is Gene Expert, and there also they got a good specificity. That is why this particular technique, the machine of TrueNat, was approved by WHO to be used in patient care and healthcare settings, and it has also been adopted by our TB program, that is National TB Elimination Program. Now, apart from these 2 tests, there are other tests, higher level tests, which are done in other facilities, especially culture and DST labs and in IRL. So once now we know that after doing X-ray, microscopy, and NAT, we have done some genetic confirmation, and one drug confirmation we have done, one first line drug resistance status. Now there is another genetic test called line probe assay. So this also has a first line drug analysis, and we have a second line drug analysis also. So first line drug in NAT we got rifampicin status. There is another drug which is isoniazid that is also tested in LPA. That is the additional information we get. And those drug resistant patients who are on treatment with these drugs, the second line drugs, so especially the fluoroquinolone class and aminoglycoside class, so those things are tested in second line LPA. Now, next we have phenotypic test. So till now, NAT and LPA are genotypic. That means you detect a particular type of gene is present or not. But phenotypic means you really take the bacteria, grow them in culture. So here liquid culture, okay, in our MIGIC technique. And after you grow them in the culture, they are again inoculated in medium, culture medium, which contains a particular drug at a particular concentration. So if that particular drug at that concentration works, the bacteria won't grow. If the particular drug is not working, the bacteria will grow in that particular culture. So we are doing the test with the real bacteria, that is phenotypic test. So currently all these facilities are not available here, and all the samples of Andaman, that is from Diglipur to Campbell Bay, all the samples are sent to IRL Chennai. So that is causing a lot of problem for us. So that is why IRL Port Blair is being established. We have finished the construction of our TB containment facility to the level of biosafety level 3. Soon we will be having these facilities of LPA and liquid culture in Port Blair itself. Now apart from this, there is another test called IGRA. Okay, interferon gamma release assay for latent TB infection. Now, all of you know that we are approaching towards our target of NTB. Okay, NTB, that is minimize the incidence of TB to great extent, and our target is year 2025. Now, all these techniques which I described till now, our X-ray, our microscopy, then our NAT test, our LPA, our culture, all these are done on symptomatic patient who comes to us, patient who come to us with symptoms, then we detect, then we treat. Okay, so by that we can decrease that number. But what about the people who have latent TB infection? So that means that they had TB infection, but after that it didn't get completely cured. That TB bacilli is dormant, is latent, is sleeping inside. Okay, so these things will get activated later when the immunity of that person is compromised. So we not only have to decrease the incident of this active tuberculosis, but we also have to find which all patients have latent TB infection, because they'll get activated later and they'll become active. So we have to find these latent TB infection and treat them also, so that the total TB incidence we can decrease for our future. Now let's discuss some basics for before we get into this PCR. So nucleic acid. So all of you know about this nucleic acid. There are 2 types. We have DNA and RNA. DNA has two strands, double stranded. RNA is single stranded. DNA, the sugar is deoxyribose, whereas RNA, it is just ribose. Then the base pairs, what we have here, we have, it is somewhat similar. Cytosine, guanine, both sides same. Adenine also same. The odd man out here is thymine for DNA and uracil for RNA. Okay, so this is the different part here. And as you can see, there is a base pairing rule. Cytosine only bind with guanine and they bind with three hydrogen bonds. And adenine will only bind with thymine in DNA with two bonds, and whereas adenine will bind with uracil here in RNA, cytosine again with guanine. So that is why you can see the color. If it is yellow here, it is green. If it is G here, it is C here. If it is C here, it is G here. That is how if one strand of DNA is there, the complementary strand is synthesized based on these rules. Now the flow of genetic information, all the important data, like if our body is a machine, it's a computer, all the data means to how to make this food, that food, everything, all small details, the information is present in our DNA. Okay, so in this DNA, all the information is present, how to synthesize each hormone, enzyme, like our thyroid hormone, though most of the hormones are proteins. So means which amino acid is used to make which particular protein. So all that information is there in DNA. That information is written, that particular information. Say we want to make a thyroid hormone. Okay, so that particular information alone will be transcribed into mRNA. Transcribed means copied. Okay, so transcribed, copied into mRNA, messenger RNA. So DNA to messenger RNA, it is called transcription. Okay. Then we have messenger RNA, and it will go to ribosomes where protein synthesis occurs. So there we have our ribosomal RNA, the major chunk of RNA, ribosomal RNA. Then we have another RNA that is called tRNA, transfer RNA. So this mRNA has brought the recipe, means what all amino acids. Because you know these proteins, this is a protein. It is made up of small blocks, building blocks, and these building blocks are amino acids. Okay, so different amino acids are there, so they have to come in certain sequence to make one protein. So that formula is brought by mRNA. Okay, and rRNA will help in synthesis of this protein. Now, what does transfer RNA do? So it will go bring all these amino acids. It will take some all the amino acids and all the amino acids, and it will bring to ribosomes. It will take all the amino acids. It will bring to ribosome to make the particular protein, and mRNA will tell which particular amino acid will go where. Okay, which will come first, which will come second, which will come third, so that we have that particular protein. And this process from mRNA to protein synthesis is called translation. Okay, transcription and translation. So before we start a PCR, we need a DNA, nucleic acid material, isn't it? So here, this is a bacterial cell. You can see the bacterial cell. Unlike us, where we have a nucleus, inside this only this DNA and all is there. Okay, so nuclear membrane is there. Then we have DNA. Whereas in prokaryotic cell, the bacterial cell, there is no nucleus. There is just nuclear DNA. Nuclear membrane is not there. So we need this DNA. So for that, this cell which has cell wall, etc., has to be broken, lysed. Cell has to be lysed and the DNA has to be taken out. So that process is called DNA extraction. Okay. So the first is DNA extraction. Once DNA extraction is done, then we move on to the main step or PCR, polymerase chain reaction. Okay. So that is nothing. The DNA we formed, now we are making photocopy, Xerox copy. Like you have one paper copy, then you want to make photocopy of 10 copies. So this polymerase chain reaction just does that. It makes multiple numbers. Then once it makes multiple numbers, then we have to detect the PCR products. Okay. So we have to detect what is this particular thing which we have amplified. Because for staining also, when you need a stain, because bacteria does not have color. So you put a stain, a color to detect. And same, say you're doing card test to see that particular immunological reaction, antigen antibody as reaction has occurred, you have to see a particular color band. So these things are colorless. So we have to visualize how many DNA we have amplified. That is PCR analysis. Okay. So that is done through various processes. So let's see what all are required in PCR. Okay, first is master mix. So in master mix, we need the one Xerox machine, the copying machine, okay, the one which makes multiple copies of our DNA, and that process is done by DNA polymerase. Okay, so the DNA polymerase enzyme is there, so that goes inside. So that is our master mix. It makes multiple copies. Then to make new copies, we need new building blocks, isn't it? To make a new house, a copy, we need new building blocks. So those are these DNTPs, nucleotides. Then other than that, we also need other components like buffer and some ions which are useful for our PCR reaction. Then we need primer mix. Now these are the things which decide which particular DNA is which we want to amplify. Because when you have a patient sample, you have so many cells in it, isn't it? Our human cells will be there. Then there will be other bacteria which is normally present. Okay. Then we have TB bacteria also. So we don't want all that DNA. We want only this TB DNA. So this particular primer, which is forward and reverse primer, it will identify specifically which DNA which we want to replicate, and it will go bind there, and it will tell where to start making copy and where to end making the copy. Okay, start and stop is decided by this primer mix. Then obviously we need the original DNA, that is the TB DNA which is present in the sample, because this is the one which we are going to copy. So that is called the original DNA or template DNA. So now the process. The process of PCR occurs in three steps. One is denaturation, then annealing, then extension. Now let's see what is this. So first denaturation. So we have say one copy of TB DNA. So DNA is double stranded, isn't it? So denaturation, what happens? It is becomes single stranded. It is double strand is broken into single stranded, and as you can see, this happens at a very high temperature, 94 degrees Celsius. Then we go to annealing. So here the temperature is reduced to 55 degrees Celsius. Annealing means to go like stick onto it. Okay, so here is where our primer will come and stick. So as I told, it will only come to specific DNA. So here are TB DNA. So it has come and stuck. So it will tell where to start and where to end. So forward and reverse primer. So next process is extension. Now it has stuck in that particular place. So now it's a single stranded. Now we have to make it double stranded. Okay. So that is where extension comes, and that is where our Xerox machine comes, photocopy machine, that is DNA polymerase. So we remember in master mix we had DNA polymerase and other building blocks, the nucleotides. So DNA polymerase will take those nucleotides, and as I told you, this base pair rule, where a particular base will attach to a particular base. So based on that, they'll extend the new strand also. So we took one double strand and made it into two double strands at the end of extension. So here you can see the temperature also varies from 94 degree in denaturation, annealing 55 degree, extension 72 degree. So now we saw in PCR, a single copy of DNA is multiplied. So by the end of the first cycle, what happens? Single copy gives rise to two copies. Okay. So number of cycles, number of copies we can decide by 2 power 1. So 1 is the number of cycle. So say most of our machines, they go to cycle 30 to 40. So say at the end of cycle 30, so it repeats this process again and again. So at the end of cycle 30, how many number of copies we get? 2 power 30. So that is nearly 10 power 9. That means 1 billion. Okay. From a single copy of DNA, you can make to 1 billion copy. So why do we need to do this? So we have to increase the number, the yield of the particular product, so that we can detect, the machine can detect this particular DNA. Okay. So theoretically it is coming to 1 billion copies. Now conventional PCR, the old PCR technique. So for that it requires a machine called thermocycler. Okay. So thermocycler. So as we saw denaturation, it has 94 degree. Then for annealing, it is 55 degree. Then for extension, 72 degree. So different temperatures. So different temperature, thermal, the temperature, cycling. So for each cycle, all these three steps are there. So for 30 cycles, the temperature go up, then they go down, then again it's raised. So all that is programmed and done by this thermocycler. So once this particular thing is amplified, we got required number of copies. Then we have to see, isn't it, the DNA what we have amplified. For that, they use different techniques. One of the techniques is gel electrophoresis. So here, based upon the weight, these whatever DNA we have amplified will be spread on this gel. Okay. Then based upon UV based dyes and UV rays, we'll irradiate it and then we'll get a picture. Okay. So this is the picture. Then we use a scale, a marker scale. So like you measure something with a scale. So like that we have a DNA ladder or DNA marker. So to tell that which particular area these things have moved and what particular weight of that particular product it will be. So say for example, we are looking for a target which has 200 base pairs. Okay, 200 base pairs. Now see sample A, B, C. Where did we get that particular target is achieved? So it is achieved here. So that sample B is positive. So now you can see it is very complicated, isn't it? First you have to amplify, then you have to do electrophoresis, then you have to read it, and there is a lot can go wrong in it, and there is technical problem. The person has to be very expert in all these things. So all these steps in our new machine, the TrueNat machine, all these particular steps, that is PCR and PCR analysis, all this is done in a single small machine. Now let's see that. So in TrueNat, DNA extraction is done inside this particular machine, that is DNA extraction machine, TruePrep machine, and these two things, that is PCR and PCR analysis, is done by this PCR analyzer. So here we have this UNO DX. This is the machine which is present in most of the place here. So here UNO means single module. So this is the single module which is present. Okay, so single module, and this is the place, a button where you have to press for this particular chip tray to open. So other than that, there is Duo, that is 2 modules, and there is Quattro, 4 modules. Now TrueNat, we have been talking about TrueNat a lot. Now what is this TrueNat? So TrueNat is a trademark, and it is marketed by an Indian company, that is Malbio Diagnostics from Goa. So based on our Make in India product, now this machine is made in India and it is by an Indian company. The other machine which we were discussing, Gene Expert, so which was already present in our program, that is manufactured by Cepheid. So that is an American company. And that machine is quite costly. It ranges near 18 to 20 lakhs. Okay, so this particular machine, machine which we saw with a single module, that costs around 6.5 lakhs. So it is little cheaper. And not only that, in Gene Expert, that for that machine, we need different site requirements, a room which is fully air conditioned because that machine will operate in a small range of temperature. Then, other than that, you need a computer which is attached to the machine. Then, you need continuous power backup for that machine. For that, you need a UPS which is 3 kVA or a 5 kVA UPS. Then, with big set of batteries for it to support 2 to 3 hours uninterrupted power supply. So, a lot of requirements are there. And in certain regions of India which are resource poor, there are frequent power cuts. There is a problem in establishing a gene expert site. So that is where this particular machine comes in. It is portable. It can be carried to field, unlike Gene Expert. And then this particular thing is battery operated. So like our mobile, you can charge them and use them. Uninterrupted power supply is not a must requirement. So this also, like Gene Expert, detects two things. That is whether the Mycobacterium tuberculosis complex, that bacteria is present or not, and also gives us idea about rifampicin resistance. Now, what is the infrastructure required for this Gene Expert? So power, obviously power is required. So as I told, uninterrupted power supply is not required, but we need enough power to charge these machines. Now coming to biosafety. So it does not require much biosafety. The ones which you use for microscopy is enough. So bench space and PPE, personal protective equipment, is required. You have your mask, your lab coats, and your gloves, powder-free gloves. Those are enough. Waste management, all the things which are used here, the tips, chips, everything has to be dipped for 30 minutes in 1% freshly prepared sodium hypochlorite solution. So you get a concentrated solution. From that, you have to dilute and make a 1% sodium hypochlorite solution. And it has to be prepared freshly every day because slowly it will disintegrate. And by next day, this will be only water. Okay, so you won't have any use. So this has to be prepared every day freshly. Then the room layout, it is small machine, so it does not require much space, but it requires 3 electrical sockets, well-grounded electrical sockets, because it has 3 machines. So you have to charge them, charge these machines. Security, the place should have a lock. Basically, it should be secure. Your lab should have a lock and key, because these are, though it is a little cheaper than gene export, but it's still in lakhs. Okay, 6 to 7 lakhs a machine costs. Ambient temperature is for our Indian condition 15 to 40 degrees Celsius. It works under humidity of 10 to 80%. Port Blair, we have the humidity is 80 or higher. So now the storage, there are different kits which come with it, how to store. So the main things which we use in the PCR part, that is chips. Okay, so there is MTB chip and there is a RIF chip. The company tells it can be stored between 2 degree and 30 degrees Celsius, but we highly recommend that you store these chips in refrigerator. Okay, because we had issues where this was stored in room temperature and we got so many invalid results. So these chips, MTB chip and MTB RIF chips, these have to be stored in refrigerator and not freezer compartment. Please don't put anything in the freezer compartment. In the normal lower part of the refrigerator, you keep these particular chips. All other things like the buffer, the pre-treatment pack, all these things you can keep in room temperature, preferably away from sunlight. Okay, away from sunlight, below your shelf or in a cupboard. You can keep these things and they have a good shelf life of 2 years. Now, how to prepare your workbench before testing? Before you start the test, obviously you put on your PPE, you wear your mask, your lab coats, and have powder free gloves. Powder free gloves we tell here because this powder can interfere with our PCR. Then you clean the work surface first from freshly prepared 1% hypochlorite, then with 70% alcohol. Anytime you clean anything with hypochlorite, it has to be wiped with some other thing because these are quite corrosive. So, instrument cleaning with 70% alcohol. This I'll talk more about it in our maintenance log when you have to do this particular thing. So please don't use sodium hypochlorite for instruments. Okay, sodium hypochlorite should not be used for instruments because, as I told, these are corrosive, and if it comes to any contact with any metal part, it will corrode and it can damage your machine. Then you should have waste containers and fill them with 1% hypochlorite solution half. Then you have to arrange all the materials because, as I told you, some of your things are inside fridge, and even say, for example, samples which had come yesterday which you could not process, that is also in the fridge. So all that has to be taken from the refrigerator and it has to come to room temperature, because all these tests have to be performed at room temperature. So take all the things from the refrigerator, keep it outside for 30 minutes, let all the things come to room temperature, then only you should do the test. Now, master checklist. What all do you need to do the particular test? So obviously, first we need is our sputum sample. Okay. Then we need our extraction device, that is prep device. So before that, to process the sample, we need a true prep kit. Then once the DNA extraction is occurred using all this, then we need to do PCR, that is our PCR analyzer machine. First, we will be using this chip, MTB chip. Then after, if MTB is detected, we will be doing rifampicin test using RIF kit. Okay. Then if you want to print out the result, you can use a Bluetooth printer. Then this transferable pipettes and the 6 microliter precision pipette, micropipette, all these are given with the kit. What we don't have is this marker pen. Marker pen you have to use to write on the LU tube, etc., the sample ID. So you need a marker pen. You need your supply of latex gloves through a powder free. Then you need our disinfectants, that is 1% sodium hypochlorite and 70% alcohol. Foot operated waste bins. Foot operated waste bins are important. And this cartridge holder PCR tube stand, it comes with the kit. So now you have to check the status of the battery. Okay, so though these don't require power supply, but these have to be charged. So they almost take 4 hours to charge, to fully charge. So if it is not charged, the test won't run. So low battery indicator, a blue color indicator will be there, then test won't run. So I advise all of you to, as soon as you come, you switch on these machines, you charge them, and when you finish your shift, 6 hour or 8 hour shift, then you can disconnect it and switch off the machine. So charging this machine is very important. Now let's see what are the parts of a TrueNat workstation. So first we are seeing this True Prep Auto. Okay, so this is the machine which does extraction. There is a buffer pack which is attached behind it, and extraction is done through a cartridge based kit. Then we have our PCR analyzer, TrueLab analyzer. So here what you are seeing is a two module machine. So you can see the two modules here. So most of the place here we have a Uno machine that is having a single module. Then we have this particular thing that is a 6 microliter precision pipette, the pipette stand. Then here you can next to it you can see PCR tube stand. Then to print the results, we have this TrueLab micro printer, a Bluetooth printer. So this can be used to print the particular result. So these are the three machines which are used in a TrueNat workstation. Now, what are the TrueNat consumables? Okay, so the first thing once you get after you get the sample, so what all things you require? So the first one is pre-treatment pack. Okay, for MTB sample. So here you can see True Prep Auto MTB sample pre-treatment pack. Okay, so this particular thing. So what all are there in this particular thing? Here you can see it is marked for 50 tests. So first you get 1 ml disposable pipettes. Okay, so 1 ml disposable pipettes are there. Then you have liquefaction buffer. So here you get 9 ml liquefaction buffer. So here you see for 50 tests, only one bottle is there of 9 ml liquefaction buffer. So there will be two times where you will be using this particular thing in your workflow process. Total per test you will be using 4 drops. Okay, so only that much you have to use, then only it will last for this 50 tests. If you pour more number of drops, then it won't last, then the rest of the kit will become a waste. Okay, they don't provide an extra liquefaction buffer. So what does it do? As the name suggests, liquefaction. So as you know, the samples, most of sputum samples, it is viscous, it is mucopurulent, so it is a little thick, isn't it? So if the TB bacteria is stuck inside, we cannot get the DNA out of it. So first, the entire sample has to be liquefied. Okay, so it has to become liquid. So that is what this particular buffer does, liquefaction buffer. Then we get lysis buffer. So this will be present for each sample, lysis buffer. So lysis buffer in the as the name suggests lysis. Okay, so once it has become liquid, the entire sample has become liquid. So it contains all the cells. It may contain human cells, it may contain other bacteria cells, and it contains TB bacteria cells also. So all the cells have to be broken because DNA is inside a cell, isn't it? So it has to be broken and the DNA has to come outside. So that is the work which is done by lysis buffer. Okay, so this is how the sample is prepared. Now we have to do DNA extraction, which is a cartridge-based sample prep kit. So here again 50 test cartridge-based sample prep kit is there. So with this you get 3 ml disposable pipette. Then you get a reagent pack. Okay, so this is also for 50 tests. So this pack goes and it is attached behind the extraction device. So we'll discuss in our next part 2 of the training, how to change a buffer pack. And this is the cartridge. Okay, so cartridge we'll be seeing how it looks. A cartridge in which the extraction occurs. So once the DNA extraction is occurred, now we need to do the PCR. So it is done in the PCR analyzer machine. So here we have different type of chips. So first one which is used in our program is the MTB chip. Okay, so here you can see the entire label is written black, isn't it? So TrueNat MTB chip. So here you can see this green color thing, SIM card looking thing. So that is our chip. With that you have a small PCR tube, and in the bottom of the PCR tube, if you notice, there is a small white color thing. It is like if you have taken homeopathy medicine, the small balls. So it is like that. So the master mix which I was telling, which contains DNA polymerase, other thing, primers, etc. All that is condensed into this small thing, ball. Okay, white ball. So that is very important. Then we have this little sensitive, so MTB plus chip. So both these MTB and MTB plus, their function is to do test and tell us MTB detected or not. So once MTB is detected, we want to know the RIF resistance status. So RIF chip. Okay, so MTB RIF chip. So this tells us about rifampicin resistant detected or not detected. These chips will be given in low quantity. So you have to be careful. You should not use this test first and then do that test. First MTB test should be done. Then only this particular test has to be done only when MTB is detected. Okay, so that is why for a LT, when he's do lot of work is there, he is doing multiple things at the time. He cannot, sometimes by mistake he'll use the other chip. So that is why you can see here the writing itself is entirely red. MTB RIF DX. So here you can see the entire thing is red. Whereas you saw in MTB chip it was black color. So this they have added benefit they have given by putting the entire thing in red, that so you realize this is a RIF chip. Now, how a sample is processed in TrueNat? Okay, TrueNat sample processing. So first, sample pre-treatment. Okay, so first you have your sputum container. You have taken the appropriate amount, 2 to 5 ml of good quality sputum. So you open the cup. So obviously, as we discussed, you have to prepare the workbench. You have to wear your PPE, everything. Then only you start doing this. Then first thing first, you have to liquefy. You have to liquefy this particular sample. So you have to put 2 drops. Okay. So you have to put 2 drops of this liquefaction buffer. Please be careful, as I told, for entire kit only 1 bottle comes. So use as per the SOP. Okay. So first 2 drops put. So notice when you are putting the drop, this particular bottle nozzle should not come in contact with the sputum cup. Okay. You should not insert it. You should keep it at certain height, little above, then only put the drop. Why? If this particular sample is say TB positive, and while bringing the sample, obviously all the edges is contaminated, and if you touch this particular sample which is TB positive, and then you do for a next sample also, so that result may get carried over, the TB bacilli will go to the next sample also and will get carried over. So most important thing, this liquefaction buffer, when you put drops, you have to put at a height, should never touch the sputum cup. So once you have put, you close the container. Okay, you close the container tightly. So then you have to slightly mix. Okay, gently mix. Don't have to shake it and all. Slightly mix so that the chemical is mixed with the sample and it liquefies. And incubate it in the room temperature for 10 minutes. Okay. At the end of 10 minutes, if the sample is not pipettable, that means it is still thick. Okay, so the sample is still thick. Then what do? You incubate it further. Okay. So the sample is still thick at the end of 10 minutes. You have to incubate it little longer. You should not add more drops of liquefaction buffer. Okay. So if you add more drops, then you lose the number of tests which you do for that kit. So incubate it for another 5 minutes, and every 2-minute interval slightly shake it. Okay. So once your liquefaction is over, now our sample is ready. It is totally liquefied. Now you open lysis buffer bottle. So this is for each sample you have 1 bottle. It contains 2.5 ml of lysis buffer. So on top there will be a rubber cork like thing, so that you discard. From the sample you have to take 0.5 ml. Okay. So 1 ml disposable pipette. So notice how he is taking this pipette. This another thing is important. All these pipettes come in Ziploc bag. So you shouldn't put your hand inside this Ziploc bag. Okay, so you open the Ziploc bag. With the help of the other hand, slowly nudge a pipette out, and you take with the other hand from outside the Ziploc bag. Please don't put your hand inside the Ziploc bag. There is a risk of contamination and you're getting false positive results even through this particular method. So please carefully take the 1 ml disposable pipette. So from here we have to transfer 0.5 ml. Okay, so 0.5 ml only you have to transfer. So in the middle you can see the 0.5 ml marking. So any pipette, the basic step is, please press the pipette fully and then only insert it below the sample column, the water column, water sample column. Because if you put it inside the sample and then you press, what will happen? Aerosol generation, bubbles will form. Then it will cause to aerosol generation. It's a risk for you. And also if there is a lot of air bubbles, it will interfere with our test. So please always press this pipette and then only insert it into the sample. Then when you release also, you have to release slowly. Please never release this particular pasture pipette or even your micro precision pipette. So never release the plunger very fast. Okay, you have to slowly release. slowly release, then fill it up to that mark, 0.5 ml mark. Okay, so this is why you have to keep this lysis buffer, the rubber bunk and all you have to throw and keep it ready. Okay, so you cannot take in one hand, then you start opening the lysis buffer. So all this have to be kept ready. So 0.5 ml you have taken. Now you transfer it to the lysis buffer. Okay, and this pasture paper discard in 1% hypochlorite solution. This thing. Now you close the sputum cup and keep the sample. See, now this liquefied sample is there. Never throw a sample till your result is confirmed. Or if you have any problem, you contact with your higher microbiologist or your TB lab supervisors, and you confirm with them if you have any problem with the result. Then for next further steps. Never throw a sputum sample till the final result is entered. Okay. So there can be a lot of problems which can go wrong. Sometimes you may have to retest, and there is a lot of troubleshooting options where you may have to re-extract. So never throw the sample till the final result is achieved. So if you are not able to do this particular test today, okay, so you put it in lysis buffer, suddenly some emergency is there, you have to go, but you have put this 0.5 ml into this particular lysis buffer, it's all right. This particular lysis buffer you can just close and leave it in even in room temperature away from sunlight till 1 week, and this DNA will be intact in it, there is no issue. So there is no problem with that. Okay, so up to this stage there is no issue. So here what happens, so again you have to use this liquefaction drop. 2 drops you have to put in this particular buffer. So here also you have to be careful that don't touch this lysis buffer bottle. So at some distance 2 drops. So total 4 drops. Okay. So first in sputum cup 2 drops, then here in lysis buffer 2 drops. So when you have very low number of tests and you don't do these tests frequently, we have found in one of our troubleshooting that the LT just leaves it in the open. So what will happen if it is exposed to sunlight a lot? This also interferes with this buffer's quality. So once you have used this liquefaction buffer, also you can keep in the normal fridge area and then use it like every other chip. So once you have put 2 drops, slightly mix and leave it at room temperature for 3 to 5 minutes. So cartridge-based DNA extraction, this is our cartridge pouch and that is the cartridge stand. So you open the pouch. So here is your cartridge. Okay, place it on the stand. So on the right side, you can see there is a black stopper. So that is where the sample is inoculated. And you can see this yellow color sponge is there. Sometime if you carefully notice on that yellow color sponge which is there, there will be a pink dot. Okay, so that is actually a processing control, internal processing control. So when you add the sample, that control will also get extracted. It will also go through all the steps your sample is undergoing, and this particular control is added so that you know the DNA extraction has occurred. The problem is if any occurs, it is in DNA extraction. The other end, you see is covered with silver foil. So once you add your sample, the elute will come at that particular part. So here you have a elute collection tube. So once elute comes after your DNA extraction, you have to transfer it to this particular collection tube. So now here you have this pipette. So elute is there. So here you can see the silver foil. So you have to use this pipette to pierce the silver foil and then collect the elute and transfer it to this particular tube. So here you can see the lower part of the pipette, there is a edge. So sometimes it is 2 parts. Sometimes if you see that particular part will be inside the pouch, you can take and attach it. So that is little in an angle so that it is useful to pierce that particular silver foil. Then you also get a label which you can stick on the tube, okay, to write the name. But I would suggest you use a marker pen and write your ID in it. Okay. So obviously first label the cartridge. and label your elute tube also. Then open cap where you put the sample. So now this lysis buffer, remember it had 2.5 ml and then you added 0.5 ml of sample in it. So total 3 ml of liquid is there in it. So that is why they have provided a 3 ml disposable transfer pipette. So now you have to be careful and you have to follow all the steps again. So press this bulb fully, then only you put it inside the lysis buffer bottle. And if you don't get the quantity fully, like till 3 ml, if it doesn't reach, there's no issue. You can do it slowly in the next step also. Like say, for example, here only you could extract 2 ml. So extract only that and try not to introduce any air bubble inside. Because if any air bubble comes inside the cartridge, it will interfere with your results. Okay. So the basic idea of this particular step is all the 3 ml of content of this particular lysis bottle has to be transferred. Okay. So here you can see slowly release, then insert it. Okay. And slowly. So here you can see just touch, don't go and dip it inside or mix it anything. Just touch the wall and slowly release it. Okay. So no air bubble should be formed. Okay. That is very important. Even if an air bubble is formed, if you slightly flick it, that air bubble will go. Okay. Now you dispose both that bottle and your 3 ml pipette. There's no use of it. Now close this particular thing. Now a DNA extraction machine, you can see. Now it is ready. Now your cartridge is ready. So preferably it is charged. So you can see the buffer count and all. So switch it on and press the eject button in the middle. Okay, press the eject button. So it will slightly open. Okay, then you have to slowly pull little further. Now you see it is like a tape recorder, like old time tape recorder we use. This is like a cassette to put inside. Now you see the orientation in which it is put inside. Okay. So this is the orientation. On your right side, the sample side is there. On the elute side is on the left side. You cannot put it in the other side and close it. So always be careful to put it in this particular orientation. Just see it is firmly sitting inside. Okay. Then push the cartridge door till you hear a click. Okay. Then you can just press the play button to start the process. So this usually takes DNA extraction 20 minutes. Okay, 20 minutes. And it slowly ejects. There will be a beep and it will eject. So here you have to be very careful. Once you load the cartridge, after that you cannot wait. If you load the cartridge, you have to do the test. Then other thing is, once you finish this extraction, you should not leave the cartridge inside because this machine is under tremendous heat. There's a lot of heat inside. If you leave the cartridge inside for a prolonged time, whatever DNA is extracted, the elute is there. Only from 3 ml of your sample, around 100 microliter of elute will be there. So if you leave it inside the machine, what will happen? That elute will get evaporated, then your entire test will fail. So slowly pull it and take the cartridge, place it in the stand. So here you can see the elute has collected. Okay, here the elute chamber elute has collected. Purified DNA is present in this particular area, around 100 to 150 microliter. Now your elute tube is ready, open it. Then with that pipette, you have to pierce it. So you can see that is slightly slanted. So pierce it. Here also, please press the pipette and then lower. Don't go inside and press and introduce air bubbles. So you transfer all the contents you transfer into this elute tube. So once this process is over, your DNA is extracted, you have put it in a tube. Say now you could only do this much. Now you have your shift has ended. There is no issue. Now this tube you store in freezer compartment, okay, of your refrigerator. All the other things which I was telling, liquefaction buffer and the chips store in normal fridge compartment lower. But this particular thing, if you want to do the next day, store it in the freezer compartment. Okay, so this has to be freezed. This has to be stored at a much lower degree. And you can next day you can keep it out and you can freeze thaw it. You thaw the whole thing, then you can do the test. Okay, the cartridge also you can discard, close it. So now DNA extraction is done. Now we have to do the PCR. So first test which we do is MTB. Okay, so here is a duo machine. So duo machine, two modules are there. Most of the places here we have Uno machine. It's a single module. So first switch on the machine by power button. So this is like a Android, like your Android phone. It takes some time to boot up, so kindly wait. So here you can see this green color light which is down. If it is low battery, it will show blue color. You won't be able to do the test. You have to charge the machine. So enter your password. Here two modules are there. That is why you're seeing one and two. In Uno only one thing will be there. Just click on the module and select the test you have to do. So here we are going to do MTB test. Sometimes in the list you will see MTB plus also. Please don't select that. In our program we are using MTB chip. So please select MTB. Okay. Proceed. Now you can enter the patient details. Okay. So referred by patient details, name, age, everything you kindly type. Sample, which type of sample? Okay. Sputum sample. For TB, we are not using blood, so kindly please don't select blood. Why that option is there? Because in this particular machine, not only TB, as you can see, a lot of malaria and even hepatitis B, there are a lot of tests which can be done. For each of those tests, separate chips are there. Okay, so that is why in the sample option, you are getting other samples also. So kindly select your sputum or extrapulmonary based on that particular sample. Start test. Once you press start test, if you have a duo module, this will automatically open. This is the place where you are going to keep the chip. But if you have a Uno module, it won't open. So next to the chip tray, there will be one place where you have to button which you have to press. Okay. So here the Uno module. So here you can see the chip tray is there. Next to that one button is there. If you press that, that will open. It won't open automatically. Please load sample and yes. Please don't press yes until you have kept the chip inside and loaded. Okay. If you put yes, then it will start, go to the next phase. So only after you finish all the steps, then only you have to press yes. So now the chip pouch is there. So chip pouch, inside the chip pouch, you have this chip, which is like a SIM conductor, like a SIM, isn't it? The golden color things are there. So this chip, please only hold it from side to side like they have shown here. You should not touch this golden connector plates, and also that white color well is there. You should not touch any of those area. Only hold it in the side. So place it here in this particular orientation. Okay, the well will be outside, away from the machine, whereas the other part will be inside. Okay, so there will be small connectors in it, so you can see hole. It will nicely sit in that particular place. Then we have this master mix tube. Okay, so here you can see that white color, that sphere is there down. Sometimes what will happen, that will be sticking onto the wall of the tube. That time you slightly flick it so that it drops down. So when you start the test, it has to be in the bottom position. That you must pay attention. Open the rubber bunk and throw it and put it in the PCR tube stand. So here you have to use this 6 microliter pipette. So it is only one volume. So you don't have to adjust the volume and all. So only one volume. So when you attach this tip also, you have to be very careful. So if you just put the pipette and just press it again. So it is in a polythene, it is in a plastic envelope. If you press, what will happen? The sharp front end of the tip, it will get bent. Okay. So only one tip is there for one test. So it will get wasted. So you have to hold the particular tip with two hands, with the other hand, then only place. Don't just press it against the envelope. Okay, so you have to be careful when you use the tip. So press it. Okay, so you have to fully press, and only 6 microliter. So here, as you saw, the 100 to 150 microliter of elute we have, but for each test we need only 6 microliter. So for MTB test, you will be doing that 6 microliter. If it is positive, you will be doing RIF test. There is also 6 microliter. So will be some troubleshooting where you may have to repeat the test. For that also, we have enough sample left. So 6 microliter, slowly release. All pipetting, slowly release. Okay. So here you have to see, go near the thing and slowly release. Okay. So please don't touch the white color thing. Slowly release along the wall, and you don't have to mix. Okay, don't keep pressing and mixing things, reverse pipetting. Don't do anything. Just release it on the side. It will go mix itself. The most important thing here is you have to keep the pipette in your hand. Don't discard the tip. There is only one tip, so please don't discard the tip. That is the most important. So many times LT by mistake he discards. Now you have to wait for just 30 seconds. So 30 seconds, please just stay there, hold the pipette with the tip in your hand. So it will, so that this master mix, the white color thing will fully dissolve. Okay, so just 30 seconds, please wait there, don't discard the tip. Then once 30 seconds is over, press it, then go below the level and slowly take. Okay, so it will take all the amount. Then your chip is kept already. Use your other hand to support your pipette hand. Okay, otherwise what will happen, sometimes there will be some shaking will be there, and you may put the drop on some other place. So no, to prevent the shaking, always kindly support your pipette hand with the other hand and just go above it. Please don't touch any portion of the chip. You should not even touch the white sample well also. Just stay above it and form a drop. Once you press it fully, one drop will be formed. Once the drop is formed, just touch with the drop. Don't touch with the pipette tip, touch with the drop. So even after drop is gone, you can see something is left. So please don't try to push it further inside. That much, that one drop is enough for the test. Now you have loaded the chip. Okay. So now only you discard the tip. Okay. So two steps tip is required. So don't discard in between. So after this only you have to press yes. Okay. So after this you press yes. The chip will be taken. When you sometimes you may have to push it. So now the test process will start. So this each chip normally it will take around 45 minutes. So you can see the real time, the temperature changes, and if you click the optical option, so you can see the each cycle change and when which control or when which sample the particular target is detected. You can see the whole process. So this occurs real time. That means every cycle, whatever change is happening, you can see real time. Okay. So click optical. So here you can see these graphs are being drawn. So you can see this. Things are being detected and the graphs is plotted. So you can see here 40 cycles have been run. So down you are seeing cycles 10, 20, 30. So nearly 40 cycles it has run. So once the thing is complete, so now you can check the result. So now it has completed 40 out of 40 cycles is over. Now you can see on the top patient ID. So just click on it to see the result. So here you can see the results. So first you see the run status, it is coming valid. So run status is valid because you remember in cartridge I was telling there was an internal control. So all that step is properly completed. So that is why you are getting this run status valid. So if there is any problem in extraction, this thing will come invalid. Okay, so once the test is valid, then only we can read the result. Now MTB, you can see MTB is coming detected and it is also giving a rough estimate of how many colony forming units were there. So it is like 3 into 10 power 4. Okay, these many colony forming units were there. And another thing you can see is CT value. So you can see the control which we used that was detected at what CT value. That means the number of cycles. So it took 26 number of cycles to detect the control, and MTB, that is our target here, the bacteria, the gene. So here it was detected at 26.33. So between 26 and 27 cycle. So it was detected. Okay, so if there are more number of bacteria already present, so it has to run for less number of cycles. Okay, so it will be detected with less number of CT value. So like 16 to 20 it will be detected. That means very high number of MTB is present. So if the CT value goes above 32 or 30, that means the chance of it being a real positive also decreases. Okay, so that you have to keep in mind. So once this test is done, then you can press the print option. So print option, it will give a printout in the printer. Okay. Or you can also press this share option. Like you have your Android mobile, from one mobile to another mobile, you can share through Bluetooth. So this picture, this entire result, you can share through Bluetooth to your Android mobile. So once it is detected, then press this MTB ref. MTB ref, if you press, then we will go to the next step. That is, using the MTB ref chip, we have to detect rifampicin resistance. Okay. So same process, same elute which we have done. So only the chip is different. So that PCR part, the chip loading part only you have to repeat. So MTB ref, you press, then you go and do the with the ref chip, and again the result you will get about the rifampicin resistance. If you check, then here rifampicin resistant detected or not detected. Here you will get the result. So once the test is finished, it is important for you to take the chip and discard it. Okay. So put it in that 1% hypochlorite solution waste bin. Please don't put it in the surface. Okay. So because these are, if it is positive sample, if you it is just lying around, that entire area will get contaminated. Okay. So you have to discard the chip in the 1% hypochlorite solution. Now moving on. So you should know that the most important disinfectant which we here use here to discard everything is 1% sodium hypochlorite. So because this is the disinfectant which we use to denature the DNA, so DNA will be destroyed. So 1% sodium hypochlorite, it should be freshly prepared every day. Okay. So all sputum samples with which has pan masala, gutka, tobacco, any food particles, blood. Okay. So all these samples cannot be processed. Okay, because these are PCR inhibitors. They'll interfere with the test and you won't get proper result. All these chips and cartridges are plastic weight and should be discarded according to our BMW, that is Biomedical Waste Management Rules. Currently, we are following 2016 waste management rules. In our health facilities, you see different color bags, yellow bags, red bags, black bags. Okay, so all that is part of this rules. So we already know for sharp and all you use this white color translucent container. Okay, so for glass you use blue color boxes. Okay, apart from this, most commonly things which are used are this black color bag. So all municipal waste, that means something which you throw and which is picked up by our municipal waste collection people. So all these cartridge cover, pouches, wrappers, the all these boxes, these are discarded in general waste, black color. Okay. So other than that, most of the things which we use in our health facility usually goes into yellow. Okay, yellow. Whatever your cotton which is contaminated with body fluid, everything goes into yellow unless that thing can be recycled. So what can be recycled? So plastics, okay, and sometimes rubbers. So all those things which can be recycled goes into red. So remember like that, all municipal things which are just covers everything of your chips and cartridges, everything, all that goes to black, municipal waste. All things which plastics which can be recycled, so your tubes, cartridges, micro tubes, elute pipettes, everything, and even your gloves. So these things can be recycled, so they go inside red. All other things like your mask, gown, and all those things, other than those made of disposable plastics, all those things goes into yellow. So it is easier for you if you remember like this the waste disposal classification. So this is very important part of any test. So kindly keep these things in mind. So now post test. Okay, so post test, I will be sending you a Google Form link to our WhatsApp group of this training. So kindly click on the link. It will open in your browser a link. So 10 questions will be there. Select the correct options. In one place, you will be asked your name. Just type your name and submit. Okay, down at the end there will be on submit option. Just submit, and I'll get your results. Okay, so you can immediately see your result also at the end of the submission. If you view test score, you can see which of your options are correct and which, if it is incorrect, what was the correct option for that particular test. Apart from this, some training documents I have attached in our Echo platform. So you can log into your Echo account, read those training documents. Okay. So next with part 2 and part 3 of this training, we'll be discussing more about how to interpret result. Like if you get one result, how to interpret it, then how to troubleshoot if any problem is there, then how to do maintenance of this particular test. And part 3 we will be talking about how to maintain registers, what records you have to maintain, and also how to report your monthly indicators which are sent to Central TB Division. Okay. So that's all for this part 1 of the training. I thank all of you for your patient listening. So goodbye for now till we meet for the next session. Thank you.